Welcome to the Wizards of Biobanking podcast. I'm Shrikant Adiga, CEO of Open Specimen, and I will be interviewing biobankers from different backgrounds and countries to learn and explore the world of biobanking. Hope you will enjoy this interview series as much as we enjoyed producing it. Thank you.
Hello everyone, I'm Shrikant Adiga, CEO of Open Specimen, and I have with me today Georget from University of Adelaide, who heads the Biobank. I let her explain her role and what exactly she does. So Georget, welcome for the second edition of Wizards of Biobank. This is a podcast that we're doing with people who are closely involved with biobanking and trying to understand both their work, their research, and as well as in general everything around what they do. So Georget, I'll let you first introduce yourself. What is your role and what exactly the University of Adelaide does?
Lovely. Thank you, Shri. Thank you for having me. It's a real pleasure. So my name is Georget Reaiche-Miller, and I am the manager of the University of Adelaide Biobank. I have been working for the Biobank since 2014, and our biobanks, all biobanks work differently, but ours is slightly different in the sense of how it came about. So back in 2011, the University of Adelaide had undergone a number of local research freezer failures, which resulted in multi-million-dollar losses. So at the time, the Faculty of Health and the Faculty of Science deans contacted the university DVCR, so the Deputy Vice Chancellor of Research, saying, we need a better solution for storing our critical samples. So what the DVCR then did was create a research or a working group, which met from from the faculties involved, and they came up with the idea of the biobank. Now, originally the biobank was supposed to be just a storage location to start with, where the samples are from, so the critical or irreplaceable material from local freezers in the local schools, the samples will be stored in the biobank. Now, that's how it all started. Now we are also work, obviously, with the faculties, but we also make the samples available for other researchers. So we communicate, create the collaborations between researchers. So the biobank has currently 48 -80 freezers. We are only storing -80 products at the moment. We are currently in expansion mode, and we're putting proposals through to include liquid nitrogen as well as -20 freezers. And also our next step would be to include processing of samples. So like I said, it started as a storage facility to support local researchers, and it continues to do so, but we're in a growing phase. We also store samples from clinical research, human samples, also non-human samples for basic research. And we also have animal samples, but we also have plant, yeasts, seeds, and soils. So our biobank covers pretty much everything.
Okay. Thanks for the introduction. So now let's, you brought up a few things, so let's get into some details around that. So one, to start with, how many researchers do you cater to?
So we currently have, we'll take one step back. Not all the researchers use the biobank, but the biobank offers other services such as helping with grant applications, helping use the LIMS, to catalog the samples in the database. So we currently have, I think it's approximately 160 plus research groups and about 54, 55 researchers actively using the biobank for storage.
Nice. So, and is this particular to a disease, or it's multidisciplinary?
It's multidisciplinary. So we have samples from cancer, we have samples from diabetes, but we also have samples from animal health. We have samples for plant and seed preservation. So we're very broad in the material that we store and the type of research that we support.
And is there, in terms of distribution, are they just spread across, or is there any one or two diseases which are like dominating the counts?
So yeah, about, from now you're testing me, but I think last time I checked, 60% of our contents were human samples, and yeah, so 60, and then we had 30% were basic animal research, and the 10% were yeast and soil. When it comes to the human research side of things, the majority is currently is for juvenile diabetes study, and also infectious disease study. So we have meningococcal B vaccine development, as well as COVID-19. You can imagine, but mainly our main users are the juvenile diabetes. But like I said, we also have cancer, we have gastroenterology, we have different areas. We also have lung disease like COPD and so on and so forth.
Is there a specific reason for that? Is Adelaide and area, more diabetic population?
No, so this particular study it's now currently an international one. So it started off as being a national study where all states in Australia were involved, both regional and non-regional, so metropolitan and regional. It's looking at juvenile diabetes. So it's the first study of its kind, I believe.
Okay.
And yeah, so they've collected patient samples from the babies, from the mothers when they were pregnant, also from siblings, the fathers. And I believe that they're now in collaboration with work in Germany. So it's a pretty big study.
Okay, so let's go into the other aspect. You mentioned that, sometime, I don't know whether it was around 2014 or when you joined, that there was an issue with safety of the specimens, or the how it was handled and things like that. So was there a specific incident, or was it in general a concern around how specimens were managed?
It was general concern around. So I did my PhD and my postdoc in virology in hepatitis B virus at the university. And at the time, our lab also went through some freezer failures, although ours was not, it was only like $1 million, nothing, more than like all the other freezers had lost. But one of the things that was an issue at the time is that researchers didn't really know what they had in their freezers. So there was no real catalogue of what contents were in there. The freezers were also not monitored or connected to security alarms. They didn't really have backup. I mean, some did. I'm not saying that they all didn't, but the most common issues that were found was the lack of security, in the sense of not connected to an alarm, not connected to backup, and researchers not knowing what was in their freezers. So if a freezer failed, then they did a backwards exercise to work out what they had in there. Had researchers known that they had really precious samples in there, then they would have taken a bit more care of how the freezers were managed. So this was found before I was part of the biobank. But what the original, I guess, findings were is poor management, really, of the freezers. Also due to funding, unfortunately, back in, I think, 2012 or 13, our National Health Research Council reduced the funding for biobanking. So there was a lot of issues in terms of what could be used for storing samples outside of out of local freezers and things like that. But one of the solutions as part of the biobank was guidelines on how to manage. So prior to the biobank being established, because it's a purposely built facility, in the interim, the first thing they rolled out was management of the local freezers. And these included cataloguing all the samples, making sure that every single sample was in the limbs, estimating, although we do not like to give a freezer a dollar value because that can change. What we've done and the way we've gone about it is that we're giving them a risk rating. So we use our university risk matrix as the basis of it. But what we have done is that we have created examples of what sample would fall under an extreme and irreplaceable category. So obviously this would be samples from patients that have passed away from a very big study that you cannot replace. It could also be, we actually have not an issue, but we have samples at the moment that were collected in the Himalayas. You can't go back and collect the same samples. It might also include samples that you no longer will have human or animal ethics to do. So those samples are irreplaceable. Then we follow down from major to moderate to minor to insignificant. So when researchers are cataloguing their freezers, their local freezers, they give their boxes and their contents a risk rating. So in the event of, I guess, in the event of a failure, but also in order to properly manage the contents, it's best to know what's really precious and what is it. Other things that were introduced were annual freezer maintenance, as well as university. So we have a procurement system where we have preferred suppliers for maintenance, preferred suppliers for university -80 freezers. We also have all the, and this is all included in the policy actually. We introduced in 2016 a university-wide cold storage management policy, where what I've just mentioned before is compulsory, and so including alarming of the freezers, having a clear emergency plan on the freezer, so who the first point of contact is if a freezer was to fail, and then also part of it in the policy states that anything or any samples that fall under the category of extreme and irreplaceable, they have to be stored in the biobank or have at least a duplicate in the Adelaide Biobank. And we strongly encourage people that have samples in the category as major to do the same. So slowly but surely, once everybody started cataloguing, ensuring that all the samples are up to date in the LIMS, then the biobank got more and more use. And then, we've started getting phone calls from interstate or other universities, saying, I know you're the Adelaide Biobank and you must have samples on liver cancer, for example. And then our role is to say, okay, I'll contact our liver research group and I say, so-and-so likes to collaborate with you. And then we don't handle those samples ourselves because we don't own them, but our job is to put them in touch so they can develop collaborations. And we've had quite a number of collaborations done that way.
Right. I think of all the biobanks that I have seen across US, Australia, etc., the focus on the safety and security at University of Adelaide is, I think, a notch above, not only in terms of the policies, the infrastructure that you guys have set up is, I think, truly amazing. Is there any kind of documentation, SOP or paper that you guys have published around this?
It's funny you should say that. I presented the work that we've done. So we are well known, I guess, for disaster recovery. And one of the things that, as you've just said, that we're really strong on is the protection of samples. And as you visited, our facility has got any backup known to man, which I'll cover in a second. But I was approached by one of the journals to put our disaster recovery plans and procedures that we have in place in a paper form, and I'm currently writing that up. So fingers crossed it'll get published and we'll be able to share that with everybody. But just in a nutshell, what we have in the biobanks is that we have all the -80 freezers connected to liquid CO2. We have set the parameters so that when the freezers get to -55, the liquid CO2 will kick in, to keep the freezer at that temperature should there be a freezer failure or a power outage. We have calculated that the amount of CO2 we have available would last us about 6 to 8 hours of extra CO2 purging in per freezer. On top of this, we also have purchased freezers in the market. And like I mentioned before, we have a preferred supplier system. We have gone to the market and looked at the freezers which have the quickest cool down time and the slowest warm up time. So technically, I think from memory, we have about 9 hours before the freezers will get to -55. So if there's a power outage or a particular freezer fails, we have about 18 hours before we hit disaster. All the freezers are connected to our university security. We have an on-call roster 24/7. I'm the first point of contact, but we also have a team underneath if I'm not able to make it or answer the call. The freezers themselves also have remote monitoring. So what we're able to see is, if an alarm goes off through security, we're able to monitor the state of the freezer. So we'll be able to see whether the freezer's gone offline, whether it's just the temperature issue, whether it's a compressor issue, and judging by the amount of temperature that's dropped, we can, assess how soon we need to attend. We also have, generally, if you look at the publications, it is recommended to have 1 empty freezer for every 10 freezers in use. But because we're telling our researchers that we are looking after protecting your extreme samples, we go a little bit further and we have 1 empty freezer for every 7 full freezers we have. So that is done so that if there's a freezer failure, we just come in, put all the contents in the same order as they are in one freezer into the next, turn it off and get the maintenance people to have a look at it. So we have all sorts of alarms in the room itself because we have CO2, liquid CO2 to the room. We have CO2 sensors, oxygen depletion sensors, water and floor sensors. So to ensure that any internal flooding or outside flooding is readily detected. So we've got sensors on the outside of the biobank. It's also built on a slight height so that if there is any minor flooding, it won't get inside. We've got pressure, so PC2, negative pressure sensors. We've got humidity sensors. We've got I think it's 38 alarm per biobank. We've got 48 freezers split over 2 facilities, and we've got 38 alarms for each one. The facility itself is only accessed by the Adelaide Biobank staff. Security do a walkthrough 3 times a day to physically make sure that everything looks okay. And we have dual authentication to enter the facility.
Yeah, that's amazing. The extent and the attention to the details that you guys have done. But one of the common question that would come up is how do you sustain the biobank with probably, I'm not sure, doing all this must be very expensive. So how do you sustain the biobank in terms of funding?
Yeah, so currently the University of Adelaide Biobank is a core funded facility. So 50% of the funding comes from DVCR or the DVCR's office, and the other 50% is split over the faculties that use the facility. So it's not costing the researchers themselves any money. So that way we're able to support them, support the researchers, ensure that the research is top quality without costing them any money. Like I said before, because the NHMRC and other funding bodies don't necessarily include biobanking unless it's part of the project, the university does its best to support the researchers by co-funding the facility.
Right. So this is something that I've seen most biobanks struggle with in terms of trying to figure out what is the best way to sustain the biobank. Is it to charge the researchers? Some people charge by collection, some people charge by storage, distribution, or in your case that is sustained. So do you think this model is much better than trying to get the researchers pay? Because I would suppose the advantage of this is that this encourages more and more researchers to actually use the biobank without having to bother about how much it will cost me.
Yes, and I have a couple of examples of that. The biobank, when it first was set up, it was on a cost recovery model. And because the grants don't run for more than 3 to 5 years, depending on what grant you got, researchers were really reluctant to store contents in the biobank because as soon as the money dried out, they didn't know what they would do with the samples, which is why the model changed for storage. Having said that, we only store contents for the University of Adelaide staff and researchers. We don't store contents for members outside of the university.
Right.
We are working on a statewide approach, and we've got a fair bit of work to do on that front. And I'm sure that if we're able to offer the services to outside of the university or include other services such as processing, that would include a fee, but we haven't made it to there yet, so I can't help you with that one. But yeah, for now, because it's only storage for the University of Adelaide staff and researchers, and it was built to protect research and ensure that top quality research is produced within the university, it is currently core funded.
Right. Okay, so let's take a break from the biobank. We'll come to this and let's talk about yourself. So you said you did your PhD in the University of Adelaide, and then you got into this. So was biobanking part of what you were doing? Like, what interested you to get into biobanking?
Absolutely. So I did my PhD and postdocs in virology, and a lot of the work that I did involved the collection of multiple. I did my PhD in hepatitis B virus, so it involved collecting a lot of samples, processing them, analyzing the data, and I particularly had a very good, being OCD, had a very good database of all the samples I had just in a simple Excel spreadsheet. I also swapped or worked in collaboration with people interstate that also knew what samples I had. And the research, unfortunately, the research in hepatitis B at the time when I finished in the lab in 2014 had reduced. Luckily there's an effective hepatitis B vaccine to protect against hepatitis. So our funding bodies didn't put it in the important basket. Therefore, the funding was, drying up, which is why the research group closed and I moved into biobanking. But the reason why I was drawn to biobanking and successful in getting the biobank role was because of my previous experience in, banking the samples for the several different research that was done within the lab. We also worked with a lot of PC2 and some PC3 laboratories, as well as quarantine material. So I had the expertise for years in this area, even though at the time the word biobanking didn't really exist, but it was all there. So once I realized that if the facility was being built to support this sort of research and seeing how biobanking had been growing internationally, I was very keen to jump on board and put my biobank hat on.
Nice. So you said you did your PhD in University of Adelaide. Were you always in Adelaide? Did you do your schooling in Adelaide too?
Yeah, so I'm originally from Venezuela in South America. I came to Australia when I was 13 years old. So I did my high school here. I actually did 1 year of medicine back in Venezuela. I soon realized that I wasn't a people person and that I worked much better behind the bench. So I came back to Adelaide, where my family was, and I did my Bachelor of Science, PhD, and stayed here. So I have actually been at the University of Adelaide since 2000. So in February this year, I would have been at the uni for 23 years.
That's nice.
In different roles, but since 2000.
And so in Adelaide, what would be your favorite thing to do?
Summer, the beach. I love the beach. You might see I'm a bit more brown than when you saw me last. Adelaide beaches are beautiful. And then in winter, I really love my hiking, bushwalking through the hills. It's lovely.
So it's you enjoy the best of both then.
Absolutely, I do.
So getting back to the biobank, you guys are not directly involved in things like participant recruitment and things like that for the researchers, right? So are there a small number of projects where you do that too?
No, so we're involved in supporting the researchers, right, but we're not involved in the research itself. So for example, one of the services that we provide is that, someone's going to put in an application for a grant, they'll come to us and say, what should we be looking at? And then we'll go through the whole process of, have you got consent? Have you got ethics? Have you got this? So we help them through the process, but we're not in charge of any of that.
Right. So, given that, then your researchers are your so-called customers, right? So those are the people whom you are actually servicing. So how do you work in publicizing your biobank to say recruit more researchers, or get more people to know what you do and how they can use
how we
services?
Can help them? Yeah, that's actually a very, very good question, because it was a bit of a struggle to start with. Even now, every now and again, people don't really understand what we do. But what I would normally do, COVID unfortunately slowed things down a bit because you couldn't, see people face to face. But what I would normally do is that I would do a roadshow twice a year, once at the beginning of the year, once mid towards end of the year. And we go through all the schools. I present what we do. I give little examples of the studies that we house, how we support them, the services that we offer for researchers. We also run a research material management training course. So one of the things that we noticed was that there was a poor labeling, poor naming conventions, poor generally management of samples. So one of the approaches that we took to start with, and I run this through my roadshows, is a little simple half-an-hour training course for any new researchers, like students, honours, masters, PhD, and research staff, to pretty much get their heads around what is the proper way of cataloguing things, what to do, what not to do. The idea behind that is to ensure that, you know, they do the right thing from day one. That way, you know, we'll not find ourselves in the same problem, you know, 2-3 years down the track. So yeah, roadshows is very important. We are resuming them again this year. I've got one in March coming up, which I'm really excited about. Yeah, so it's pretty much going out there, going to all the schools, ensuring that they understand what we do, what services we offer, and letting them know that we're here to support them with a lot of things. So not just biobanking, because not all researchers will need to use the biobank facility, but they will use our support.
Right, and these are typically you know, like individual sessions you do in different places,
yes,
or is it
so
like
it's like a seminar. It's like a seminar series sort of thing. So what I would normally do is that I would book a seminar room through the different schools on different days, and I'll do an hour and a half session, depending on the questions and how many people show up and how they're interested. So we normally run maybe 5 or 6 in the first half, and then again 5 or 6 in the second half. And the turnout varies. Generally, our busiest time is the March, because more people are coming in and studying at the university. Our mid to end year is not as busy, but it's still good. It still has a good turnout.
Yeah, that's a pretty interesting you know, approach, because the more you do such things, even though I mean it's more of general education that you're trying to impart, it also directly or indirectly publicizes what you do,
Yes.
for people to know
Yeah,
that you exist.
one of the things that we exist and that we support them, but one of the things that we have had good feedback on is that there have been a lot of different researchers able to collaborate with us, like being the middleman. So, for example, there has been a couple of groups that have been after particular cell types or particular diseases or particular controls, that they would reach out to us and say, I am after this. I know you came and did a roadshow. Do you work with any of these particular samples? And then I'll be like, yeah, we do. Such and such researcher has these, and then they get in touch. And we have actually had very good feedback from that. And like you said, you know, making researchers know that we're there and what we do has opened the doors. A lot of researchers didn't know that the same or similar research was being done on a different campus or something like that. So it's been quite helpful.
Right. Do you record these sessions and put it on your website or something?
No, but that's actually a really good idea.
Yeah.
I guess now with COVID, there's the research material management course I did record, and there's a PowerPoint recording of that, but I haven't actually thought about recording the roadshows, but I might actually do that.
Yeah, I mean it is just free content, right? So
yes,
there's not much effort in doing that, but then you put it on your website, it's there for people to look at it and throughout the year.
Yeah, that's a great idea. Thank you.
So in terms of you know, if I had to ask, like, what are the challenges that you face in your in a general biobanking operations? What would be the, you know, top 1 or 2 things that would come to your mind?
Yeah, so like you mentioned before, we're pretty lucky in the sense that funding is not a problem for us at the moment. The 2 main issues are staffing and space. So right up until mid last year, the biobank was just me. I managed it. Angus Netting's the director. He's supported the biobank since he adopted me. And the biobank started falling under Angus's role in 2015. But I didn't have any support. So we do freezer audits, local freezer audits, as well as audits for the biobank, transfers, helping, supporting researchers writing grants and all of that sort of thing. just fell under me, as well as, usual maintenance and that sort of stuff. Luckily, mid last year, we have a biobank technical officer that supports me. So yeah, staffing is a big issue, and the other issue is space. So luckily, the University of Adelaide has been very lucky in the sense of getting being funded multiple grants. So there's been quite a few increases in research groups, new schools coming on board, or new institutes coming on board, which means that they increase, or the demand for biobanking has increased immensely. And running out of space to support our researchers is a thing. So, if you ask me what my top two would be, space and staff.
Are you looking at off-site storage, long-term off-site? Because if I remember correctly, all your freezers are in the main campus, right?
Yes, so we are in the main campus. We are currently in the process of looking to have a purposely built facility off-site, but that's in very early works. We don't know exactly where that's going to be or how big it's going to be, but it will be purposely built for, hopefully, like I said earlier, -80s, liquid nitrogen and processing, so the whole lot, but it'll be a little while yet.
And any plans for certification for the biobank?
Yeah, so that's actually one of the things that we discussed at ABNA this year with NATA. There's a lot of different aspects of our biobank that would be approved through the new ISO, including risk management. At the moment, we're not thinking of officially having it accredited, but what we've done, and what we always do, is to ensure that our biobank meets the ISBER guidelines for biobanking. In some areas we've gone above and beyond, as you mentioned before, but we're also trying to work towards the accreditation. So we look at every single point of the ISO, and we try to have it to standard. So in the event that a particular researcher needs the facility to be accredited, or if we decide that we want to be accredited, the work won't be too much because we're already working towards those standards.
Nice. So your main, other than providing these services for the researchers, your other goal is to match the demand and the supply, right? So people who need specimens and people who have specimens, how do you do the matchmaking? And one of the so what are the ways that you do that? And a related point to that is one of the things that others have been doing, like we saw in ABNA, people like University of New South Wales who showed their specimen catalog that they had built using Openspecimen. So would those kinds of tools be of use when you're finally online with Openspecimen?
Absolutely, and that's one of the features that I'm really looking forward to using from Openspecimen. So the UNSW Biobank, I really love the way that you guys have set up the catalog. It's something that I really like to do. Like I said, at the moment, people just contact me out of the blue and say, do you happen to have this? But if we actually have a catalog available to researchers, then it's going to be a lot easier and more productive for the researchers themselves.
Yeah, it makes things easy, as well as makes things easy for, say, writing grants, so they don't have to contact you and ask for 100 things when things are available readily online
on the page. Yeah, no,
yeah.
Absolutely. I was really excited when I saw that feature on their website, and I'm like, yep, that's something that we'll definitely be using.
Right. So anything else that we missed talking about in terms of the biobank work before we move on to other things?
No, I don't think so. I think, like we all know, each biobank is different. They're all special in their own different way. I think it's just really important that researchers know what the value of the samples. I mean, not the values in the dollar value, but as in how important the values are to making a difference. And it doesn't necessarily have to be human samples. It could be plants, it could be animals, it could be anything. But having a facility that can support them in improving their research and providing, support and services, I think it's really crucial. And it'll be great to see more of this, not only in Australia, but, worldwide, and collaborating amongst the researchers and biobanks is key.
So that brings up a good segue into what I wanted to talk next, which is value of the specimen. And the value of the specimen is also kind of directly linked to what kind of data you have about the specimen, right? Because otherwise
Yes.
it's just, someone crudely put it that otherwise it's just meat in a freezer. So,
True.
so, unless you have, good quality data associated with that. So how do you manage that, especially given that you are not really in charge of collecting, these specimens are managed? Do you kind of guide your researchers in terms of data collection, minimum data points
Yes.
that they can collect and things like that?
Absolutely. So we have a guideline of minimum information that researchers should obtain for each of their samples. And we provide this to all the researchers for the local freezers. I can't enforce how much information they put in for the local contents. However, the fields that must be filled in for samples to be stored in the biobank. So we got mandatory fields that must be completed. And when we do a transfer, we ensure that all the minimum information, I guess would like to call it, that's all filled in for our biobanking purposes. There are a lot of research groups out there that have the funding, I guess. That's the other thing. So a lot of local researchers might only be 1 or 2 staff, might have only 1 or 2 staff. Others might have 14, 15 staff. So some actually have the time and able to dedicate someone to update the information for the samples, others not so much. So we provide them with the backbone information, and it's technically up to the researcher or the research groups to use that to their advantage. But in terms of biobanking, yes, we have, your data is only as good as you put in. So you won't be able to know as much what you have unless you put the information in. So yeah, we definitely have like a template of minimum information that is required for prior to storage.
Right. And then as part of the package or the biobanking service that you provide, one of the services you also provide is that they get to use the LIMS, that central LIMS that everyone can use. So that
Correct.
also makes it kind of a good deal for them, where they don't have to deal with their Excel sheets and things like that.
Exactly. So when this was originally rolled out, we went from some groups that had no idea what they had in their freezers. Some groups had yellow Post-it notes on their freezers. Some had some printouts, others had Excel. So there was complete variety of cataloging or knowledge of what they had in their freezers. So when we introduced cataloging into the LIMS as the policy, the majority of the researchers were incredibly grateful that they were able to have a system that they can import the data and know, update it and know exactly what they have.
Right. Okay, and that's so that kind of talks about the biobanking stuff that you have been doing. The other thing that you have been very actively involved is the ABNA association. Can you give us an idea of what is ABNA? Maybe starting with the full form of ABNA,
Yes.
and then what's your role, and in general, what does ABNA do?
Sure. So ABNA or ABNA is the Australasian Biospecimen Network Association. I had a mental blank then. We are a non-for-profit organisation. We support research or researchers that do biospecimen science and that are actively involved in banking. We have been around for 20 years this year, so make sure you come and celebrate. We run a yearly workshop or conference that normally runs for about 2 days, 2.5 days, 1 day. We're slowly growing. Originally, it developed from ABN Onc, which was ABNA Oncology. It was very cancer focused. It became more broad. It didn't only include cancer, it also included other diseases, human diseases. And in the more recent years, we've been working to be a more inclusive society, so that we also have biobanking knowledge, biospecimen science knowledge, share that information amongst the different fields. So human, animal, plant. What we have found is that there's a lot of areas that we share in common, but yet we're not aware of. So, we have things like ethics or consent in the human front, but you also have ethics, and in some areas consent for animals. There's also a consent for plants, depending whether they're native protected species. So there's areas which spread across the different fields of the different specimens that we work closely to, to ensure that we're all supported. So one of the things that ABNA is really proud of is the networking and the collaboration. So as you experienced last year, for example, we always have, COVID put a bit of a downer, but we still managed to have an online meeting, which was great. But the face-to-face interactions, the number of collaborations and publications that have come out because of the networking between the different biobanks. ABNA works really strongly to ensure that Australasia itself, so it's not just Australia, it's Australasia, to ensure that we all work together. There's no point in reinventing the wheel. So currently ABNA also has subcommittees where people are able to express their interest and work in areas that they really know about. So, we'll have a publication subcommittee, we have a disaster recovery subcommittee. So we have different areas or different areas of resources that members would and actually use in their favor. So I think it's an amazing, platform for collaboration, networking, improving biobanking, not reinventing the wheel, using support and resources readily available to everyone. So it's great. And this year is our 20th anniversary. We're having our 20th celebrations, in the Gold Coast. We're at SeaWorld this year, so please feel free to come along again.
Yeah, all the more reason to attend the conference. So
absolutely.
And you conveniently sidestep the fact that you are the vice president and the
oh yes, I forgot that. I'm currently the vice president, and Cassandra Griffin is the president. And this year in October, when the conference is, I step up as president. So I'll be a bit busy, but I'm really looking forward to it. ABNA, so the committee members are an absolute amazing to work with. We all have really great ideas to bring on. We have lots of members which contact us all the time with ideas and support. So they're a fantastic team. So although I'm a little bit scared, I'm really looking forward to it too.
So, and the conference is typically in October every year?
Yes, so I can't remember from the top of my head. 18th, let me have a quick look. I believe, if I'm not wrong, I don't want to get ahead of myself, I better check the date. So yeah, from the 18th until the 20th of October this year. So it's normally towards the end of October. So we're at the Gold Coast. We are planning to have a really, really good program. Like I said before, being inclusive, introducing different areas. So one of the feedback that we had from ABNA in WA in Perth last year, can't say last year, it's only a few weeks ago, that they really enjoyed the fact that they were involved in the process, not really understanding that they were biobankers. So we had animal scientists of, biospecimen science in animals as well as plants, and they would get up there and say, I didn't realize I was a biobanker. So, and that really resonated with us, and it's something that we're definitely going to, improve and keep growing. And
at a very basic level, probably anyone who has a freezer and specimen is a biobanker.
Correct. But people, when you think biobanking, people just think of a big room with a freezer. No, if you're looking for a particular study, a particular disease or anything that you're collecting samples for, you are a biobank. So it's really exciting to see people understand what they're doing, and how it can all be part of this family, I suppose, that we can all help each other.
Yeah, one of the in the Perth conference, one of the most interesting session that I attended was the one on koala. I don't know whether I'm pronouncing it right.
Koalas? Yes.
Koalas. Yeah.
Yes.
So, yeah, very, very interesting, different things that otherwise you don't see in your normal day-to-day work.
Exactly. I have learned so much from other areas of research that had not, that I wouldn't have even thought of. So it's fascinating.
Sure. So that pretty much brings us to the end of this interview. I do plan to get in touch with you again next year. Maybe once you had an year full of using Open Specimen, we can talk in more detail about
absolutely
about how you are doing with Open Specimen. So on the Open Specimen side of things, recently you decided to start using Open Specimen, and since you're not really live with Open Specimen, I will not go into that in detail. But in general, from an IT infrastructure point of view, a question that I had was, what was the process you went through in terms of, if I remember correctly, you went through a tender process, and you.
Yes.
So how did you go about doing that? If someone is planning to do something similar, what would be the things that you tell them?
Exactly. So the first thing you need to do is to really understand what you're going to use the LIMS for. Is the LIMS going to be used for a biobank alone? Is the LIMS going to be used for university-wide, like we're using it, as well as the biobank? Understanding what you want to get out from the LIMS. Do you just want to have a freezer catalog and that's it? Or do you also want to have a, something that will let you track the workflow of a particular sample? Do you want to know what samples were generated from what parent, and so on? So understanding what you actually want to get out from the LIMS, understanding your budget. So there's a lot of different LIMS out there. The prices are completely different. The offers of maintenance on a yearly basis is also completely different. So work out how much money you're able to spend, what you're really after, and make sure that, well, this is what we did anyway. So when we went out to tender, we had particular categories that we needed addressed. So each of the applicants would address those points. We then had a panel, and I can't remember how many of us were there. I think it might have been 10 of us, where we all then read through the application, saw what suited us, what didn't suit us. We had, the university provides you with a scoring sheet. So each particular category had a value. We all give it a value. So all the people in the tender, in the group, gave it a value. Then we pretty much went through the process of who was the top, who was not even, look at. You then go back to those particular vendors and say, look, I really like this feature. I see that you don't have this feature, but how, talking to the vendors is absolutely important. I remember I chatted to you, you were able to give us a demo, which was incredibly helpful. It's just understanding what you're after and asking the questions is key. So you guys are, answered so many questions for us, and you were able to give us a demo of what we would look like. So that's really key. Understanding what you want from the LIMS, how you want to use it, the amount of money that you're willing to spend upfront and on a yearly basis. Make sure you have particular categories of questions, and also ask the vendors the questions that you're after.
And two quick questions on that. So how long did the process take? Did you do any?
What was that? Sorry?
No, I said the second question was, did you do any site visits to other centers to look at what they do with their IT infrastructure?
Yes, yeah. So because during COVID, and we didn't physically do site visits, but we do have through ABNA, actually, we have a lot of collaborations with different banks throughout Australia. It was pretty much of what system do you use, what do you like about it, what do you recommend, what you don't recommend. And that's actually a very good point there, Sri. If you like a particular product, go out to sites. If you can't physically go to sites, contact them and find out how they're using it, whether they're happy with the system, what's the pros, what's the cons. So that's pretty much what we did. Once we narrowed down the applicants, we then contacted the different groups and said, I know you use this, what do you think of it? I know you use that, what do you think of it? Nothing is better than actual word of mouth and experience.
Yep. Yeah, the reference checks that you do also gives you, it's not just the software that you're behind, right? You also need to look at what kind of support, things like that. So
that makes a world of a difference. You might be stuck doing something, or something doesn't work. If you're at the end of an email, or at the end of a call, answering and supporting the researcher, that changes everything.
Absolutely. So that's all pretty much that I wanted to cover.
Thank you, Sri.
Anything that you think that we missed?
I think you've covered everything. And like you said, I would love to have a chat to you once we've actually gone live with Open Specimen and see how we go. But I'm really looking forward to it.
Sounds good. So thanks, Georget, for giving us this time. As I said, the goal of this is not really around talking about Open Specimen or its features and things like that, but more educational in biobanking world, especially because every biobank is different, and then
Yes.
trying to take out the uniqueness of those biobank and help others understand what you're doing. Maybe there are a couple of things from that people can get inspired and try to improve their own processes. So it was really useful from that point of view.
Absolutely. And like I say, there's no need to reinvent the wheel. If someone's already done something, collaborate with them. That's what we're here for. Work together, give them ideas. It's great. So I really like this podcast that you guys are doing, and thank you so much for asking me to be a part of it.
Thank you so much.